Namaskar. Today we are going to study about the sandwich ELISA. So we have already studied ELISA in detail. We have also discussed the difference between direct ELISA and indirect ELISA. Then today we are going to study in detail about the sandwich ELISA. And what is the advantage of sandwich ELISA over direct ELISA? So just to recall, in direct ELISA, the antigen is in direct contact with the antibody which is conjugated with the enzyme. In indirect ELISA, the antigen is not in direct contact with the antibody which is conjugated with the enzyme. That is, they are in contact through a primary antibody which may be the antibody present in the sample. So in sandwich ELISA, what we do, the antigen is sandwiched between two antibodies. Okay, one antibody is the capture antibody which is bound to the microtiter plate. This is the capture antibody which is bound to the antibody microtiter plate. Then we have the detection antibody, and this detection antibody, if it is conjugated with the enzyme, then directly we can add the substrate and it will be converted to a colored product and we can analyze. Or we can use this detection antibody, we can use an unconjugated antibody, that is the primary antibody. Then we will add the secondary antibody which is conjugated with the enzyme. Then we will analyze. So sandwich ELISA can be two types, direct and indirect. So let us see. So in the sandwich ELISA, the antigen is conjugated between two antibodies, and one antibody we call it as capture antibody, the other antibody is detection antibody which may be conjugated or unconjugated. But if it is unconjugated, then we have to add another antibody, that is secondary antibody which is conjugated antibody. So if it is unconjugated one, if this antibody is unconjugated, if this antibody is conjugated one, then if this antibody is conjugated one, then it is direct because we can add the substrate and we can observe the change in color. And if it is unconjugated one, then first unconjugated antibody, then we have to add the secondary antibody. Okay, so two antibodies we will use here. So one is unconjugated antibody followed by conjugated antibody. So two antibodies if we use, then that is indirect. So this sandwich ELISA detects antigen. So it is more specific than the direct ELISA. So in direct ELISA, the antigen is in direct contact with the conjugated or detection antibody, and in this sandwich ELISA, the antigen is sandwiched between them, and that detection antibody can be conjugated one or unconjugated one. If it is unconjugated one, then we have to add another secondary antibody called conjugated antibody. So it has advantages like high specificity, high sensitivity, very small amount of sample is required, but disadvantage is longer protocol, challenging to develop. So let us see direct sandwich ELISA. So the microtiter plate or microplate first coated with a capture antibody, So suppose this is the microtiter plate well. So first we'll have the capture antibody coated with the capture antibody, then wash with PBS buffer, unbound capture antibody will be removed, then we will add the blocking agents like BSA. Suppose these are the blocking agents like BSA, so the space are blocked now. Then we will wash with PBS to remove any blocking agent. Then we will add the sample which is containing the antigen. Suppose say this is the antigen. This is the antigen. So now if sample has the antigen, it will bind. Then we will add another antibody, and that antibody, if it is conjugated with enzyme, then it is direct, and if we use one antibody which is unconjugated, then secondary we have to add. So in this case, this enzyme will change when we add the substrate. It will change into a color product. So wash with PBS, then add the sample contained antigen, wash with PBS, add detection antibody conjugated with the enzyme, wash, add substrate, then we will have either a color product or no color. So if it is called showing color product, the color is changing, then that means antigen is present, sample is positive. If there is no change in color, then sample is negative, antigen is absent. So this is direct sandwich ELISA. The antigen is captured between the capture antibody and the antibody conjugated with the enzyme. And in indirect ELISA, the same thing, the same figure. This is direct. This is for direct sandwich ELISA. The same figure, suppose we show it here. Same figure. The capture antibody, then wash, then so we can add the surface blocking agent. The surface we can block so that no antigen or antibody can bind here. Then we'll add the sample containing the antigen, so it will bind here, the sample containing the antigen. Now to this, if we add the antibody which is not conjugated, this is one step. Then we have to add a secondary antibody which is having affinity with the primary antibody. So these are the primary antibodies. Then we add the secondary antibody conjugated with the enzyme. So now we can detect. So this enzyme will convert the substrate to a color product. So this is indirect sandwich ELISA. So this is the difference, and in both the case it is called sandwich because the antigen is sandwiched between the capture antibody and the detection antibody. This is all about. So indirect sandwich ELISA, microtiter plate, add or coat the capture antibody, then washing with PBS, add blocking agent, then washing, add sample or serum containing antigen, capture antibody binding will be there, then washing, add unconjugated detection antibody, that is the primary antibody, then washing, add conjugated detection antibody, that is the secondary antibody, then washing, add substrate. If there is colored product, sample is positive, antigen is present. If there is no color, sample is negative, antigen is absent. So this is all about sandwich ELISA. Thank you.